Um, Hi everyone, my name is Layan and I'm a third year medical student. And today we're going to be covering the microbiology crash course. So I know that you guys have a different doctor this year, but overall it's the same block director. So inshallah it's going to be the same block objectives and same things that you're going to be tested on. So I'll be covering the gram-positive, the gram-negative, and the non-staining bacteria or atypical or other clinically significant. Okay, so it's going to be 6 lectures in total, so it is going to be a very long session. So feel free to join on a specific lecture that you have trouble in, not necessarily attending the entire session. But okay, so let's start. In the previous session, I hope you guys covered the differences between gram-positive and gram-negative. This I'll be going through it very quickly. So in gram-negative, the main difference is that we have an outer membrane and we have a very thin cell wall, the peptidoglycan cell wall. Okay, now this is an important concept to understand so that you're able to remember the difference in colors between them when we stain them. So in first step in staining, we use a violet stain or like a bluish stain. Okay, so it's a violet bluish stain. And when we stain it, it's going to stick in the cell wall, the peptidoglycan cell wall. Then we use iodine. Now it's important for you guys to know the role of iodine, which is the fixation. It basically puts or keeps the color in place. It forms complexes with the dye so that the dye sticks in its place. Then we use alcohol to wash away the dye. Okay, now this is where the main difference happens. In the gram-negative, because it has such a thin peptidoglycan cell wall and it has a very thin outer membrane, the dye gets easily washed off. Okay, and even it loses its outer membrane from the alcohol, and the dye in the cell wall gets washed off because it's a very thin cell wall. Okay, but the gram-positive, it holds on to the dye. Okay, so now when we use the counter stain, which is pink, or the fusion stain, only the gram-negative is going to appear pink or red, while the gram-positive, because it already had the bluish-violet stain, it still appears bluish-violet. So this is the main thing to differentiate, is that the gram-positive appears blue or purple, and the gram-negative appears pink or red. Okay, now a mnemonic for this can be, that I use to remember, is PB, which is peanut butter. So positive is always blue. Okay, even if it's purple, it means there's blue. You know what I mean? But if it's pink or red, it means no blue was involved, so it's negative. Now let's start with the first lecture, which is the gram-positive bacteria. Now, the way we classify gram-positive is from the shape. So we either have the cocci or the bacilli. Now these are round, okay? So they look like little balls. For the bacilli, they're rods, so they're like sticks. Okay, so this is the main difference. Now, remember we said PB is the mnemonic, so positive blue, peanut butter. So if it's gram-positive, it's going to appear blue on gram stain. So all of these that you see here are gram-positive, okay? Even if you see like something that's purplish, always go with gram-positive. This is very important concept to carry on like through all of the rest of the blocks from at school, because even in year 3 and the end of year 2, some people were getting questions wrong regarding this in other blocks because they didn't go through POD really well. Okay, so have this concept really well in your mind. Even if you have a hint of purple, it means that there is blue dye that stayed, which means it's positive. Okay, any hint of blue or purple is positive. Now, cocci means round. Okay, so anything that's round, it's going to be called cocci. Okay, uh, now how do we basically classify the cocci further into staph and strep? So staph is clusters, like you see over here in this picture. They cluster up, okay? They form clusters. While strep, they form chains. Okay, they see how they're chains. They're not really groups, okay? Like this one, for example. Okay, they're chains. A way to remember this is strep, as in like, you know how kids line up to go on a trip, you know? So strep trip, so remember that it's in chains, okay? So again, the way we classify gram-positive is based on shape. So we either have the round ones, which are the cocci, and we have the rods or the bacilli, which are like sticks. Now, how do we classify the cocci further? It depends on if they cluster up or they line up. Okay, so the clusters are called staph and the chains are called strep. Now, these are like my notes from last year, but it's really important to have a very good foundation. So Remember how we said we classify the round ones based off of if they clump up or cluster, or based off of they line up, which is staph and strep. Now, this is morphologically speaking, shape speaking. What if we want to chemically know if we have staph or strep? I don't want to have to look at the shape to know are they lining up, are they clustering up? I want to do it just a chemical test to know what am I dealing with. So the test for that is catalase. This is extremely, extremely important. It's very high yield. It's definitely going to be asked on. It's the first differentiating step in cocci, okay? So it's catalase test, okay? So if it's catalase positive, it means we're dealing with staph. If you have catalase negative, it means you're dealing with strep, okay? So this is how we differentiate between staph and strep, okay? Now, I'm going to be focusing on staph for now, then we'll move on to strep, okay? So staphylococci, it's very easy. Cocci, we said it's round, and staph, we said they cluster up, or in other words, it's catalase positive. How do we classify staph further? We're going to be talking about this. Okay, so let's start. Now we're going to be focusing on the staph. Okay, so we have three types of staph: staph aureus, epidermidis, and saprophyticus. We're going to be starting with aureus first. Now, how do we classify staph? An important thing is coagulase. Coagulase is the most important thing you need to know. So if you have an organism that's catalase positive, it means you're dealing with staph. How do you know which staph exactly you're dealing with? We look at the coagulase test. Okay, so again, go back to the notes. We said that it's catalase positive, so we're dealing with staph. How do we classify staph further? Depending on if it's coagulase positive or coagulase negative. Okay, if it's coagulase positive, it means we're dealing with staph aureus. This is very, very important. Almost in every past question, past paper, you'll see that question that goes like, we're dealing with a catalase positive and coagulase positive organism. Which of the following is it? Staph aureus. Okay, because it's the only one that has catalase and coagulase together, so it's staph aureus. Okay, now let's talk about staph aureus in detail. Now, anything where I go into detail, it means it's very, very high yield. Like everything is high yield about it. Now, for staph aureus, I got a lot of requests to focus on agar and virulence factors. So this is what I did for all the organisms. Now, basically, what's a virulence factor? Virulence factor, it means it's a special characteristic of that pathogen or organism that makes it cause the disease. Okay, for example, if it has coagulase, it means it can cause little clumps in the body or blood. If it has an exotoxin, it means it can release toxins, which can lead to the consequences. What if it has a capsule? Okay, organisms that have a capsule are extra protected from the immune system, which means they can stay in your body for longer and reach more distant organs. That's why you'll see in other blogs and even in this blog that any major diseases like meningitis, which is inflammation of the meninges of the brain, or for example, for the lungs, like anything that reaches these really distant organs, really important organs, they probably are capsulated organisms because they were able to kind of like stay in the immune system. They were able to evade the immune system. Okay, so virulence factors, they're basically special characteristics of this organism that make it cause the disease that it causes. Okay, that's why they're important to know because by understanding the virulence factor, you're kind of able to connect the dots on what's the disease that it causes, you know? So again, anything that has super antigens, then it's very likely that it causes septic shock. Okay, so let's go through them. Now, how can you remember these? I need like mnemonics for this. So basically, you have the capsule, okay? A way to remember this is CCC, okay, for the Staph aureus. Coagulase, we can remember this because we said they cluster up, you know. Catalase, we said this is very obvious. It's catalase positive because it's staph in the first place. So anything that's staph is going to be catalase positive, and it has a capsule, and it has protein A. A for aureus, that's how I remember, AA, okay? And a really, really, really important aspect of the Staph aureus is that it releases exotoxins, okay? So it releases enterotoxins and which act as superantigens. Okay, now superantigens are very, very, very virulent, and what they do is they directly attack your immune system. They don't go like, the normal process you took in immunology where antigen presenting and then it calls up the T cells come and they recruit and they attack. No, it avoids all of this. It just directly causes intense proliferation of the T cells and it really, really causes an aggravated immune response. Okay, so these superantigens lead to toxic shock syndrome. Okay, which is like you have like it's like a septic shock. Okay, and the enterotoxins, entero from enteric, so GI. Remember that it causes food poisoning. Okay, and it also causes hemolysis. Hemolysis means heme is blood. Lytic or lysis means it breaks down something. So hemolytic means it breaks down blood cells. Okay. And an important thing to know is that it has beta lactamase, which means um, it's resistant to the antibiotics. Okay. Did you take beta lactams, which are like the antibiotics in pharma? It's basically able to break those down. Okay. So yeah, so again, what's the mnemonic? Basically, remember Oreo or Oreos? Oreos sounds like Oreo. So remember, it causes food poisoning. And A for A protein. And then a mnemonic to remember the conditions is stay silent to play first. So S is for sepsis. So don't forget the toxic shock syndrome because of the superantigens. And the other S is for skin. So skin conditions like the scalding of the skin, anything like all skin conditions. When you see skin conditions, Staph aureus is on the top diagnosis differentials. Remember that it's toxin mediated. P for pyogenic means it causes pus formation. Okay, like in this case here, you can see pus accumulating under the skin. And F for food poisoning. Okay, now on microscopy, we said that PB, because it's positive, it's going to be blue. Okay, peanut butter. So positive means blue. We said that it clusters. So you see it in clusters, okay, not in chains. And we said that it's round. Okay, so they look like little balls. Okay, so this is how you identify Staph aureus. And for the agars, now you probably took this in lab, which is basically an agar is like a thing you grow the bacteria in, okay? And it helps you differentiate different bacteria from each other. So we have different types of agars. They can either be differential or they can be selective, they can be just nutrient, they can be enriched, okay? Each type of agar has a specific job. For example, a really important agar to know is the mannitol salt agar. Okay, mannitol salt, it means it has two things. It has mannitol, which is like a sugar, and it has salt. Now, this makes it selective and differential. What do we mean by that? Basically, the fact that it has salt selects only staph and it inhibits the growth of every other organism. So the only thing that's able to grow on the mannitol salt agar is staph. Okay, whether it's staph aureus, staph epidermidis, staph saprophyticus, all of the staph can grow on salt while all the others are inhibited. That's why we call the salt aspect of mannitol salt selective, okay? Because it selects stuff and it like inhibits the growth of any other genus, okay? The mannitol aspect is differential. What do we mean by differential? It means it can differentiate different types of stuff. Okay, mannitol is not selective because it does not inhibit a certain type and allow another type to grow. So it's not selective. It's only differential, which means it can differentiate different types of stuff. Now let's go into detail. So if we have mannitol salt agar, it means we're dealing with stuff, right? This is point one because we said everything else is inhibited. Point number two is that an important characteristic of Staph aureus is that it ferments mannitol, or it's able to break down mannitol. Okay, when it does that, it turns yellow. Okay, why? Because we have also a pH indicator that's attached. So when it breaks down the mannitol, things are released, which changes the pH, which causes a color reaction. No need to know all this. Just know that the salt inhibits everything else but the staph. How do we know if we're dealing with Staph aureus or Staph epidermidis, Staph saprophyticus? Like which type of staph are we dealing with? We look at the mannitol fermentation because aureus does ferment mannitol and it does break it down, it causes this color. Okay. And an important way or a nice way to remember this is, do you know what aura is? Aura is like this color that's like surrounding something. It's like this bright, like, illuminescent color. So that's how I like to remember. It's very like golden and very like it looks like an aura. So a positive mannitol fermentation is yellow. Okay, remember this. So this is to know if we're dealing with Staph aureus. Now, this is blood agar. Blood agar basically allows it's an enriched agar, so it just allows the growth of many, many, many organisms. They just appear differently on each. Okay, it doesn't inhibit growth. It doesn't really do anything. It's also differential because by looking at the organism, you're able to see how it grows. Okay, I'm not sure about the differential part. I don't think it's differential. I don't know. I'll double check and let you know. But just know that by growing different species on the blood agar, you're able to tell what is what. For example, Staph aureus looks golden. Okay? Like we said, it's like an aura. So it has like this golden pearls to it. Epidermidis are white, very, very, very white. Okay? So just by growing it on the blood agar, you're able to see what kind of organism you're dealing with. Okay? Now, like we said, we also have, we said that staph is catalase positive, which is what differentiates it from strep. Okay, so this is the catalase test, which means that we basically can tell if we're dealing with staph or strep. Okay, if bubbles form, it means there was a reaction. Okay, which means we're dealing with staph. But if there's no bubble formation, it means there wasn't a reaction. Okay, so this is probably strep. Okay, if you're like trying to differentiate staph and strep. But this is catalase negative, so it can be any other organism than staph. Okay, so basically we said coagulase. Coagulase can differentiate different types of staph. So if you do have coagulation, okay, you're dealing with staph aureus. If you don't have coagulation, you're dealing with the other type of staph, probably epidermis. Okay, so we're done with aureus. And do you guys have any confusion? Because I can't really see the chat. Any questions regarding aureus? You guys can unmute if you want, whatever you want. Okay, all good. Okay, so the other types of staph are very easy, nothing much to know about them. Just the very high yield thing you need to remember is that epidermidis, it has like an extracellular slime that helps it stick on catheters. So if you have a question that's like, which organism is associated with catheter infections or catheter insertion infection? It's mainly the epidermis because when you insert a catheter which, into the body, okay, the epidermis is very sticky, so it tends to stick to these things. So anything like that's foreign that you enter into the body, like for example, if you have a knee implantation, which is metal, these kind of things, foreign objects in the body, it's mainly staph epidermidis because it has a slime that causes adherence to the devices, or catheters. So this is also a very repeated question. I don't think your doctor emphasized it though, so I'm not sure. Now, saprophyticus, just know that it's for urinary tract infections in young women and cystitis. High yield. Okay, this is like a picture that shows it when you insert catheters. Okay, the epidermis gets excited and it's like it wants to cause the infection because it sticks to it. Okay, so yeah. Okay, let's quickly review staph before we move to strep. How do we differentiate staph from strep? If you want to unmute or type, no problem. Catalase, yes, exactly. Now, if it's catalase positive, it means we are dealing with staph. And if it's catalase negative, we're dealing with strep. Now, what are the different types of staph? Okay, we have saprophytic, or saprophyticus. What else? Epidermidis and aureus, exactly. Okay, now how do we differentiate the different types of staph? We know that staph in general is catalase positive. Now, how do we know exactly which staph we're dealing with? I want like a chemical test more. Coagulase. Exactly. Now the agars, it's that's very true. We can also use the mannitol salt agar and coagulase. Okay. Which agar is selective for staph only? You guys already mentioned it, the mannitol salt agar. Okay. Now, can you list some virulence factors for Staph aureus? Protein A, exactly. Remember the mnemonic. So A for protein A, super antigen, okay. We also said Staph is catalase positive, so catalase is also uh, a virulence factor. We said it's coagulase positive, so coagulase can be a virulence factor. Exactly, CCC, catalase coagulase capsule. And the superantigen and beta lactamase. Yes, exactly. You're all correct. Now, um, let's move on to strep. Okay, so these are the different types of strep. Now, like we said, we differentiate staph from strep by doing catalase. Now, if it's catalase negative, it means we're dealing with strep, not staph. Okay, now we have a different way of classifying um, okay, before we go into that, yes, so strep is positive. So again, peanut butter, P is blue. Okay, but this time there, strep means chains. Okay, they're not in clusters, they're in chains. Remember, the kids are taking a trip. Okay, they're walking a line to take a trip. So strep is chains. So you can see very clearly that this is strep. Which type of strep? We don't know, it's just strep. Okay, so how do we classify strep? It's a different classification system than the staph. So for staph, we said we classify based on coagulase. For strep, we classify based, do you guys know? Based on what, how do we classify strep? Yes, exactly, based on hemolysis. Now, again, I'll repeat this over and over again. Is strep catalase positive or negative? Negative, exactly. Now, how do we classify strep again? It's based on hemolysis. Again, what's hemolysis? It's basically lysis means breakdown and heme is blood. So how do they break down blood cells? Okay, or to what extent do they break down blood cells? That's how we classify strep. Okay, now we have alpha hemolysis, we have beta hemolysis, we have gamma hemolysis. Each one is a different type, okay? Obviously. Now there's a mnemonic for alpha hemolysis, which is GPA. A is for alpha, P is for partial hemolysis, and G is for green. Okay, now alpha hemolysis is partial breakdown of the red blood cells. Beta hemolysis is complete breakdown of the red blood cells. Gamma hemolysis means there is no breakdown. Okay, so it doesn't really show anything. Okay, so the partial breakdown of red blood cells, it's because they produce hydrogen peroxide. Okay, these organisms that are alpha hemolytic, they produce hydrogen peroxide as a byproduct of growth. So as they're growing and metabolizing, they produce hydrogen peroxide. What does this hydrogen peroxide do? It starts breaking down certain parts of the red blood cell, not the entire red blood cell, just certain parts of the red blood cell. That's why we say partial. What are these things that they break down? They can convert hemoglobin into methemoglobin. Hemoglobin, oxygenated is red in the blood, and when it's converted into methemoglobin, it becomes green. Okay, that's why on the blood agar, if you put the alpha hemolytic organisms on blood agar, they're going to show up as green because they convert hemoglobin into methemoglobin. Okay, now I think I forgot to attach a picture for this, so I can show you guys in the end, just remind me. But like on the agar, this shows up as green. Okay, GPA, remember that. Now, the two organisms that are alpha hemolytic are strep pneumonia and strep viridans. How do we differentiate these? By the Optocian test. Optocian is basically an antibiotic, okay? And not every organism is sensitive to it, and not every organism is um, resistant to it. So if we put Optocian on the organism and it's sensitive to it, you're gonna see like a ring around the organism where like nothing is happening, you know? And if it's aptotion resistant or the opposite, sorry. So if it's I'll show you a picture later. I'll explain on the picture. I'm sorry, the confusion. But like this is how we differentiate on if it's pneumonia or verdance. We do the aptotion test. Okay, aptotion sensitive means it's pneumonia. Aptotion resistant means it's verdance. Okay. Now moving on to beta hemolysis. These are the most important ones. Basically, beta hemolysis means it's the complete breakdown of red blood cells. Okay, complete breakdown of red blood cells. Why? Because it has um, okay, I'm not going to go into too much detail. Let me just stick to this. So it has complete breakdown of red blood cells, complete breakdown. Okay, so it appears as bright yellow on the blood agar. Okay, and the main characteristic of the beta hemolytic strep organisms is that they contain exotoxins and streptolysin. Okay, now the two that fall under this category is Streptococcus pyogenes and Streptococcus agalactiae. How do we differentiate between these two? It's the bacitracin test. Okay, so for group A or alpha hemolytic, we use aptotion to differentiate between them. For group B, we use bacitracin. This is very convenient because the alpha sign looks like an O, so aptotion. Beta hemolytic for B, bacitracin. Okay, so to differentiate alpha hemolytic, we use aptotion to differentiate between beta hemolytic, we use bacitracin. Okay, this is just not important. No. Uh, yeah. So now to start with the alpha hemolytic, we said alpha hemolytic, we have pneumonia and viridans. Okay. Now the yeah, so the most important one is pneumonia. So this is the one I'm going to focus on. Now this one has a polysaccharide capsule. Okay. Now this is very important because this is used in the development of like vaccines and stuff. Okay. It also has IgA protease. Who knows where IgA is present? Like, where do we have the majority of IgA in the body? Yes, body fluids, and you took this in immuno, you should know this, I think. On mucosal surfaces. Okay, so IgA is present in body fluids and mucosal surfaces. So if something has IgA protease, you can kind of tell where does it cause infection? Where does it cause the bacterial infection? So it causes pneumonia, meningitis, and bacteremia. Now, mucosal surface, you can tell it's the like the respiratory tract, you know, that's why it causes pneumonia. And it can also go to meningitis. Who remembers why this organism is able to cause meningitis? I mentioned this in the beginning. It's able to reach the brain because it's hiding from the immune system. The capsule, exactly. So the fact that strep pneumonia causes meningitis can help you remember that it's capsulated. Okay, because this helps it like stay in the body longer and reach more distant organs and infiltrate more. Okay, and the fact that it has that it causes pneumonia can help you remember that there's IgA protease. Okay, these aren't directly related. I'm just trying to give you like a way to memorize them, how I did, you know. And what else? Yeah. Now there's a vaccine for strep pneumo. And do you guys know what serotypes are? They're basically just like different strains of the same organism. So different, for example, we have strep pneumo, okay, but each organism can have like a different kind of polysaccharide on its capsule. But it's still strep pneumo. We're still dealing with the same organism. It's just each one we have different strains. Okay, so the vaccine for the strep pneumo covers multiple serotypes. Okay, and an important thing to keep in mind as a concept in general is that the immune system, like let's say you get infected with strep pneumo, that is serotype 7, your immune system only forms antibodies against 7. It does not form against 13 or 23. It only forms like basically the immune system is serotype specific. Okay, so if you get infected by this type, uh, but then you form antibodies against it, so you're protected against it. But the next time, if you get infected by strep pneumo 13, you'll also get the infection again. Your body is not protected from the different strains. That's why COVID was very hard to make a vaccine for, because each one had a different strain, you know? So it was very hard to find a vaccine that covers all of those strains, you know? Now, this doesn't cover every serotype, but it covers like a good few. So it's a very good vaccine. So what are serotypes? Again, they're different like polysaccharide arrangements on the capsule. Okay, so this is very important to remember. Strep pneumo, P polysaccharide capsule. Okay, so yeah, so a way to remember this is PPP. So remember that strep pneumo has a polysaccharide capsule. It has IgA protease and it has pneumolysine. Okay, so yeah, again, how do we differentiate it from strep viridans? Do you guys remember? It's alpha hemolytic. So what does the alpha look like? Aptotion, exactly. Aptotion sensitivity. Aptotion sensitivity can help you differentiate if it's strep pneumo or strep viridans. So strep pneumo is aptotion sensitive or resistant? Sensitive, exactly. And viridans is aptotion resistant. So this is how we can tell the difference between them. Uh, this is a very important picture, and these white parts basically show you the capsules for the organism. And the strep pneumo comes in diplococci, which means it's two circles stuck together. Okay, so this is easy to remember because strep is chains. So just remember that pneumo is two. They're just chains of two. Okay, so any questions for alpha hemolytic? Okay, inshallah, it's all clear. So just remember PPP. Remember the vaccine for the polysaccharide capsule? Remember Aptotion? Okay. Now, remember the beta hemolytic ones? These beta hemolytic ones have their own unique classification system. Okay. So the beta hemolytic strep can be further divided into group A and group B. Okay. Why do we do that? Because they noticed that the organisms in the beta hemolytic organisms, they have specific antigens on their wall or their membrane or their capsule. They have specific antigens or carbohydrates that are very specific to that organism. So they were like, okay, let's classify them, you know, because all of the pyogenes have specific cell wall carbohydrate antigens. Okay, and all of the agalactia have specific cell wall carbohydrate antigens. So we can easily group them into A and B because these have A antigens, these have B antigens. You know, they have very specific antigens specific to that organism. So that's why they classified them. The classification system is called Lancefield. I don't know if you took the exact name. This no, okay. So for group A, we have strep pyogenes. For group B, we have strep agalactia. Now, a way to remember this is the galaxy is black. Okay, so remember agalactia is group B, beta hemolytic strep. Okay, so it's group B, beta hemolytic. Okay, and yeah, okay. So I always like to remember this before this, you know, because when I remember this, I go like, oh yeah, Diogenes is A, you know, because this one is B. So they cause very, very different kinds of diseases and they have different virulence factors. Again, what did we say about virulence factors? They're basically characteristics of the organism that help it cause the disease, you know? So whether it's enzymes that it has, whether it's the capsule, whether it's just any characteristic of the organism that helps it basically harm the body. Okay. And I'm not gonna, like, I know there are like so many virulence factors for each organism, but I'm only mentioning the ones that are very specific to this or very like relevant to the disease, you know? Because if I give you a list like hyaluronidase or this, all the enzymes that it has, like that's not, you don't really need to know all of this, you know? Just know the ones that are very relevant, very high yield, and they're directly related to the disease that it causes, you know? So let's start with group B. I like group B. So Agalactia, again, galaxy is black. So always remember that this is group B, beta hemolytic, all of these, you know? And it's capsulated. How do you remember that it's capsulated? We said you can remember this as like, how do you go to space? You sit in a little capsule and you're sent to space, you know? So I don't know, you can yeah. So the fact that it's capsulated, you can kind of like predict what kind of disease it causes, meningitis. Okay. So That's why the capsule in eGalactica is very high to remember, you know? For example, group A is also capsulated, but it's not that stressed on. You don't really find that like point of discussion regarding to pyogeny that it's, oh my God, it's capsulated, you know? No, that's it's basically like just related to the disease. So group B, it's very important to remember it's capsulated because that's gonna take you to the meningitis, okay? And it's also CAMP positive. Now, this is very important to remember. CAMP is basically a test we do in the lab, but it's not mentioned anywhere in your slides. That's why I'm not gonna go into it. But remember that Agalactia is CAMP positive. If you guys watch Sketchy, which I cannot stress enough how helpful Sketchy is. You're gonna remember how the mnemonic that was given for camp, just basically the baby sitting in the little camp on the moon in the galaxy, if you remember that. Yeah. And there's like a helmet on the baby, which will remind you it's capsulated. If you watch Sketchy, you'll know. So, an important thing about agalactia is it causes neonatal meningitis, and it's part of the normal vaginal microbiota. So, basically, as the baby is being born, natural birth, not C-section, it has to be natural birth, it's going to be exposed to this normal microbiota, which can lead to neonatal meningitis. Okay, now going to group A. Group A is very, very important, and basically, okay, the main valence factors you need to know are protein F, M protein, and streptolysin. Okay, so basically, this is a very important condition, especially in CVP. You're going to take it next year. So let's start one by one. Let me go back to enter. Okay, first thing, it has protein F. Protein F, if you can see here, it's for adhesion purposes. So it helps the bacteria or the pathogen adhere to the cell it's trying to invade. Okay. Protein M is used for evasion. Who knows what's the meaning of evasion, not invasion? Evasion. Do you guys know? Okay, I don't think so. Okay, evasion means it evades the immune system. Okay, so evades or like you can say avoids the immune system, you know, or it can hide. It's very cunning protein. So what it does is, um, yeah, so what it does is it prevents opsonization. You guys took opsonization in immuno, but if you didn't study immuno yet, that's okay. Very crammable, so don't worry. Basically, opsonization is the way that you tell the immune system, come attack the cell. Okay, there's a foreign cell in the body. It basically attaches a marker to it that helps the immune system recognize this cell as, okay, I need to attack this cell. So the M protein, it prevents that from happening. So it can hide from the immune system, okay, because it prevents the opsonization. So it prevents the complex system from attacking the cell, okay? So I'm not going to go into immuno details in case you guys didn't study it yet. So the M protein is very, very important to remember. If I want you to remember anything from this, it's the M protein, okay? Now, we'll get to why also this is important. Now, in the beginning, the group A causes impetigo and erysipelas and pharyngitis. Okay, the main one is pharyngitis. So remember as skin conditions and pharyngitis. It also causes scarlet fever. Now, the important thing to remember is in the end, after you've been infected and everything, your body makes antibodies against the disease. This is a very general concept, right? So what the body does is it recognizes the M protein and makes antibodies against it in the long term. Okay. Now the problem is this M protein, it's very similar in makeup to the myosin cells of the body. Okay, to the proteins on the myosin cells in the heart. Okay, so what the antibodies start to do, they start to attack the myosin cells of the heart. So this leads to rheumatic heart disease. Okay, so in the beginning, when you're first infected by this organism, you get pharyngitis and like these skin conditions and scarlet fever. Okay, then after the infection, over the long term, the body creates antibodies against the M protein. So your body has these antibodies. So after a long term, your antibodies start to mistake the myosin cells for the M protein and it starts attacking the heart cells. This is called molecular mimicry. because the myosin cells are mimicking the M protein, so that the antibodies start to attack it and you get rheumatic fever or rheumatic heart disease. Okay, so this is a very important concept to remember. Streptolysin O and also important. So this is what the condition looks like. Another organism that also causes the skin conditions is basically, do you guys remember which mnemonic had skin in it? Yes, Aureus. Stay silent to play first. Yes, so Aureus also has the skin conditions, okay? So remember that these both cause the skin conditions. So firstly, you get this, you get scarlet fever, you get pharyngitis, okay? Then over the long term, you get the antibodies attacking the myosin cells and you get rheumatic heart disease, okay? Is this concept clear? Okay, so this is basically a summary of the what we discussed. So first thing, catalase. Catalase positive means staph. Catalase negative means strep. Okay, staph is clustered. Strep is in chains. Remember, strep kids are taking a trip. Okay, so staph is classified based on coagulase. Staph aureus, is it coagulase positive or negative? Positive, yes. Now, strep, on the other hand, is classified based on hemolysis. So we have alpha hemolysis, beta hemolysis, gamma hemolysis. Now, the alpha hemolysis are classified based on aptotion sensitivity. So the strep pneumo, is it aptotion sensitive or resistant? Yes, it's sensitive. And the viridans, resistant. Okay. Now, we're done with with the alpha hemolytic. Now, the beta hemolytic, they're classified based on cell wall antigens. So we classify them into group A and B. Okay. Now, another way to tell between them, I don't know if I mentioned this. I did, right? Bacitracin. Don't forget this. Okay. Beta hemolytic B to bacitracin. Okay. Yes. Okay. So the beta hemolytic, they're classified based on the cell wall antigens. So we classify them to group A and B. Which one is group A? Exactly. Which one's group B? Remember, galaxy is black. Okay, yes, agalactia. So, what color does alpha hemolysis look on blood agar? Remember GPA. Green, exactly, because there's partial hemolysis. What color does the beta hemolysis look or give? No, it's yellow. Wait, did I say black? It's yellow. Black is just the mnemonic. I'm sorry if I made you think it's actually black. It's the mnemonic. I just remember that the galaxy has the color black. So that's why I remember that Agalactia is group B. It's not the actual color. Okay, just to emphasize. Beta hemolysis is yellow. Okay? Okay, I'm sorry for the confusion. Okay, so now we're gonna move on to the bacilli. We were done with the cocci, which is um the round ones. Now we move on to the rods or the bacilli. So this is the second lecture, I think. Yeah, this one is way less dense. Okay, this one is very easy, inshallah. So these are the three gram-positive bacilli: Clostridium, Corynebacterium, and Listeria. Okay, so let's start. How are bacilli classified? They're actually classified based on spore formation. Okay, so spores are basically, I think you took it in Dr. Hanat's lectures, which is basically this thing, they're like layers around the organism that help protect it against like extreme environments, against temperature. It's like a little shelter that it helps the organism survive against adverse conditions. Okay, so we have spore forming and we have spore non-spore forming. So spore forming is Clostridium and Bacillus. So I don't know if this shows the spores. I think there was another better pictures. But anyways, so these are gram. Is this positive or negative? Positive, yes. Remember peanut butter? So peanut for positive and butter for blue. Okay. So the spore forming are the Clostridium. This is the important one. Non-spore forming is Corynebacterium and Listeria. I wanted to include the Corynebacterium picture here just to avoid confusion. Do you see these red dots? These are not spores. Okay, so if you get this picture, please don't put this as like Clostridium. These are not spores. These are like granules that they're basically chromatic granules, and they're very specific to Corynebacterium. Okay, so these are granules, they're not spores. This is like a special stain for Corynebacterium to show these granules. Okay, now the difference in shapes, the non-spore forming are more like they're short and chunky. Okay, so if you want to remember it like that. Now, this one I just copied it from the doctor's slides because they're very general. I'll also use Ambos to summarize it. For Clostridium, we have four types. We have perfringens, tetany, difficile, botulinum. Now, tetany, you can easily remember that it causes tetanus, okay, which is like spastic paralysis. Your muscles start contracting, and like very spastic paralysis. Difficile from C, you can remember it causes colitis. So it's mainly GI related. Okay, colitis, colon. Okay. T for tetanus for T, tetani for tetanus. Okay. And perfringens, I like to remember that it causes food poisoning, but I remember like perfringens kind of sounds like fridge. I don't know. So yeah, it also like very important concept is that it causes gangrene and infections. Like basically, this is very commonly found in soil. And when you cut yourself or you have an injury, it's very common that you get infected by this organism. If it's like if you're outdoors and you don't clean the wound. So it's really good at penetrating deep wounds, and causing like necrosis and gangrene. Botulinum basically causes flaccid paralysis, which is you're not able to move at all. Okay. Now this is a really, really nice summary from Ambas. Um, can you guys see the top of the screen or is it covered? I don't know. Oh, okay. So let's start with tetany. So tetany basically it causes tetanus by the release of exotoxins. Okay. And perfringens, as we said, it's found mainly in the soil. Okay. And gastrointestinal. Remember, fringin sounds like fridge, so it also causes food poisoning. Okay. So it's found in the soil and it's really good at causing gangrene, which is like really deep necrotic infections. It's mainly found in like deep wounds. Okay. And difficile, C from for colitis. Okay. So it's also GI related and it causes pseudomembranous colitis. Okay, which is in the colon. How does it do that? Through toxin A and B. I'm sure there's no really easy way to memorize these. It's just like through repetition. Okay. Botulinum is found in canned foods. This is also important, like hint in questions. Okay. Uh, yeah, that's it. Now for Corynebacterium, which is non-spore forming, a really nice thing about this, or very unique thing, is that it has metachromatic granules. Okay, which are these red things. I also added a picture here from Google because it's very cool. Um, if you zoom in, you see these little granules, which have which are very like intense in color. Okay, so yeah. Now, Corynebacterium, there are many types, but the main important one that you're usually tested on is diphtheria, Corynebacterium diphtheria. Now, this one just know that it causes diphtheria from the name. How does it do that? Through the diphtheria toxin. Okay. How does the diphtheria toxin work? It basically inactivates EF2 or elongation factor 2. Okay. Elongation factor 2 is needed in protein synthesis. So if there's no protein synthesis in the cell is just going to die. Okay. So again, Corynebacterium diphtheria, it's the non-spore forming gram-positive bacilli. And what it does is it produces the diphtheria toxin, which inhibits protein synthesis by inactivating elongation factor 2, leading to cell death, and it causes diphtheria. This is what diphtheria looks like. Pseudomembrane means it forms like a layer over your mucous membrane, okay? Forms like a layer almost, okay? So pseudomembrane, pseudo means it like it's almost something, but it's not really something, you know? So pseudomembrane, it forms something that's kind of like a membrane, you know? So this is what it looks like. This is the diphtheria pseudomembrane. And an important thing to remember is the DTaP vaccine. D is for diphtheria, T is for tetanus, and P is for pertussis. Okay, so the T is for tetanus, which is basically the Clostridium tetani, and D is for the diphtheria, which is Corynebacterium diphtheria, and P is for pertussis. We're going to be talking about that later. Okay, so do you guys any questions regarding the bacilli? Okay, okay, okay. Now I'm gonna just add some questions to make sure that gram-positive in general is understood because we finished gram-positive. So let's go to the first question. These are all from pasts, by the way, so they're very accurate questions. Okay. If an organism is coagulase positive and catalase positive, then what is it most likely to be? C. Okay. Oh, you were answering. And okay. Yes, it's Staph aureus. Okay, great. I'm glad you guys know this. Now, which of the following is gram-positive diplococci? Okay, someone said A. Okay, what is B? Is it gram-positive or negative, this picture? Yes, it's positive. Is it strep or staph? Strep, exactly, because they're in chains. What about C? What is C? Is it positive or negative? Positive, exactly. Strep or this is also cocci positive or cocci positive. Cocci just means circles. Okay, so these are also circles. So cocci does not really help us, but like, is it staph or strep? This is staph, exactly. Good question. Okay, so just a reminder, uh, gram-positive, we differentiate based on shape, cocci and bacilli. The cocci can be staph or strep. Staph is in clusters, strep is in chains. Okay, now this gram-positive, negative, because it's a very bright pink or red. Okay, so you're right, it is A. Who can tell me what organism did we say is gram-positive and diplo, if you remember? Pneumonia, exactly. So this is strep pneumo, okay? Because they're also in chains. It's just a chain of two, you know? So yes, so it's pneumo. What structure is the pinpoint? I didn't show you this before. I wanted to put it in questions. If you can guess. That's exactly, these are endospores. So could this be Clostridium or Corynebacterium? Exactly, Clostridium. Because Corynebacterium is non-spore forming. Okay, so yeah. Which infection is associated with medical catheters? Exactly, epidermis. Okay, any questions regarding gram positive? Anything you want to ask on? I'm not gonna, Akira, you didn't cover every single detail because your lectures are very, very dense. So I try to focus on like main things. Okay, okay. We're gonna do a few slides and take like a prayer break. So gram negative. Gram negative also we classify based on shape. We have the cocci or cocci, doesn't matter, bacilli and cocobacilli. These basically are like they're not circles and they're not rods. It's like a mix of both, you know? So we're gonna start with the cocci. Okay. So cocci is basically Neisseria. Okay, I know there's also this one, but you guys didn't take it. So I'm just gonna cover Neisseria. Okay, so Neisseria is the gram-negative cocci. Okay, so we have two types of Neisseria. We have meningitidis and gonorrhea. Okay, so there are general features of Neisseria. They're not specific to meningitidis or gonorrhea. They're just features of Neisseria in general. Okay, so these are the features. Basically, as you can see, they're bright pink. Okay, pink or red. Okay, which means it's gram-negative. Okay, not positive. There's no hint of blue. Okay, so pink or red is gram-negative for both, of course. Now, a special feature about them is that they look like coffee beans. Okay, if you look at this here, they really, really look like coffee beans, you know? And another feature about them is that they like to live inside the white blood cells. Okay, you don't just find them floating around. Okay, they like to be inside. Not, they don't have to be, but they like to. Okay, so they're found inside leukocytes. So I think you guys took HLS. So you can tell that these are the white blood cells. Okay, so this is the Neisseria, not this entire thing. Okay. Just in case you cramped HLS and have no idea what's happening, these are white blood cells. And the thing inside, these little coffee beans are the Neisseria. Okay? Now, what else? An important thing is that these have a special agar that's used to grow them. Okay? It's selective to the Neisseria species. Only Neisseria can grow on this agar. It's called Thyre-Martin. Okay? A way I like to remember this is I just say Martin went to Syria. Okay, so Thayer Martin, Neisseria. Martin went to Syria. Okay, so remember Thayer Martin is selective to Neisseria. Which agar was selective to Staph aureus? If you guys can remember. Yes, mannitol salt agar. Yes, the mannitol part was selective to Staph aureus. No, the salt part was selective to Staph. Okay, good. Uh, Yes, so Thyromartin is selective to Neisseria. It's diplo, so you'll find them in twos mainly. And they grow on heated blood agar. Now, there's a difference between the Thyromartin agar and heated blood agar. The Thyromartin is selective, okay? So we literally only use it if we're suspecting Neisseria, okay? The heated blood agar, it's called an enriched agar. What does enriched agar mean? Enriched agar means that it's basically enriched with nutrients that this organism needs to grow. Okay, so it's not really selective to Neisseria. We use it in many other organisms. We're going to go through them. But basically, when you heat blood, the blood cells break or they lyse. So that releases certain nutrients or factors within the blood cell, making like providing the nutrients allowing the bacteria to grow. Okay, so heated blood agar is also called chocolate agar because it turns brownish. Okay, so again, when you heat the blood, it breaks the red blood cells, releasing nutrients, and these nutrients allow the organism to grow. Okay, because the Neisseria needs special nutrients to allow it to grow. Doesn't mean it's selective. No, it's just an enriched medium. Okay, so we can grow it on heated blood agar, and in order to confirm it, we use the Thyromartin agar, because it's very selective. And an important thing to know is that it has protease IgA. So again, because it has protease IgA, we can remember or we can guess what kind of diseases it can cause. We can guess that goes to mucosal surfaces, which can be the genitoureteral tract, it could be GI, it could be respiratory, we're gonna see. We can tell that it has an adherence to um, mucosal surfaces, okay? Because if you destroy IgA, it means there's no protection for mucosal surfaces, so it easily invades those areas, okay? Now, let's start with Neisseria meningitidis, okay? So Neisseria meningitidis is capsular. Okay. So because it has a capsule, again, what can you guess is the disease that it causes? Meningitis. From the name, this is very easy to remember because there's meningitis in the name. And in order to cause meningitis, you want to take a break for prayer? Sure. Let me finish this one and then we can go take a break, okay? So basically, because there's meningitis in the name, okay, you can tell that it's capsular because we said in order to invade such a distant organ like the brain, in order to reach that, you need to have a capsule. So this is the main virulence factor, okay? Um, Another thing to remember is that it ferments maltose and glucose. Okay, now this is very easy to remember. Just remember Mg, M in meningitis, and G. Okay, so M and G. M for maltose fermenting, and G for glucose fermenting. Okay, this is also an important thing. Okay, now how is the Neisseria meningitidis spread? It's actually spread by droplets. Okay, what do I mean by droplets? Like coughing, sneezing, any droplets. That's how it's spread between people. And basically, if it affects the brain or the meninges, where do you think we can find this bacteria sitting? In the CSF, which is the cerebrospinal fluid. It's the fluid basically surrounding the brain. Okay. And again, we have these are really important. Okay, this is very high yield and we'll probably be asked about it. Okay, Neisseria meningitidis has different serotypes. Okay, which means in a different strains, as we explained, they're A, B, C, Y, W135. Okay, you need to remember these. Okay, you need to remember A, B, C, Y, W135. Okay, and it's oxidase positive. This is the oxidase test. Okay, so if he gets you this picture, or he gets you an organism is oxidase positive and is suspected to cause meningitis, for example, what is it? What's most likely the organism? Neisseria meningitidis. Okay, so it can either cause meningitis alone, or sepsis with or without meningitis, or meningoencephalitis. Okay, meningo means it affects the meninges, which are like the outer layers of the brain. Encephalitis means it affects the actual brain itself, not the coverings of the brain. This is like you don't need to know, it's just if you want to tell the difference. So again, Neisseria meningitidis, we can tell it's capsular because meningitis. If it affects the brain, you can find it in the CSF. MNG, remember that, maltose and glucose. And basically it has droplets. Okay, we can like pause the recording so you guys can have a prayer break. Okay, so we stopped at the Neisseria meningitidis. So, as we said, just quickly, because I feel like I rushed it, a way to remember the main virulence factor is remembering that it causes meningitis from the name, to remember the capsule, and a way to remember that it's oxidase positive. How I like to think about it, it has nothing to do with like the actual reason. It's just a way to memorize it. Is that the brain is it's the most damage when it loses oxygen first. So this helps me relate it to oxidase, so I don't forget it. Again, nothing to do with the actual reason. It's just a way to remember it, oxidase. Okay, because it's very important. It's a very characteristic feature of Neisseria meningitidis. Okay, and yeah, so that's it. Now for the second type of Neisseria, um, again we're talking about gram-negative cocci, okay, which are the Neisseria in general. So the second type of Neisseria uh, is actually a sexually transmitted disease. Okay, the main virulence factor here is IgA protease. Again, because we have IgA protease, we're able to know that it has an adherence to mucosal surfaces. Okay, so what does this mainly cause? It causes urinary tract infections, okay? And the genitourinary tract in general, okay? It also causes neonatal conjunctivitis, okay? Remember, the eyes is also a mucosal surface, and eye infections, urethritis, cervicitis. So all of these are mucosal surfaces. So you can remember IgA protease, gonorrhea. It's important to know that it's sexually transmitted. Okay. And a special thing about Neisseria gonorrhea is that it prefers low oxygen, like a low oxygen environment. So we actually supply it with carbon dioxide to grow it. Okay. And again, it has a good drug resistance. And the way gonorrhea develops drug resistance is by two methods, either through beta-lactamase or through permeability, and it decreases the permeability of the antibiotics. So the antibiotics are not able to penetrate through the microorganisms. Okay, so again, we can't really tell if this is Neisseria gonorrhea or meningitis, but a clue in the scenario is going to tell you. For example, if they told you we found Neisseria in urethral exudates, urethral, of course it's not going to be meningitis. If they told you we found it in the CSF, you're going to know and it's probably, okay, Neisseria meningitis. So this is a summary. Again, remember, it can be grown on chocolate agar, which is an enriched medium. Okay, it has a
Excuse me,
selective.
Layan.
Yes, sure.
Somebody in the chat is asking you to repeat the permeability.
Oh, sorry, I didn't see that. Okay. So, in order for the beta-lactams or antibiotics in general, in order for them to uh, have an effect on the microorganism, they need to penetrate the cell. So if we're reducing permeability, the antibiotic is not able to reach this microorganism and kill it. Okay, so this creates drug resistance. The organism becomes resistant to this drug because it reduces its permeability. Is that clear? Okay, no problem. This was emphasized in your slides, I think. It had like its own part. So just remember, beta-lactamase and permeability is like the two main methods it uses to develop drug resistance. Now, this is the main summary, and these are the main general features of Neisseria in general. So remember Thyromartin, Martin went to Syria. Okay, so this is very selective for Neisseria species and or genus. The chocolate agar is an enriched nutrient agar. So again, what is chocolate agar? It's just heated blood. Okay, we heat up the blood breaks or the blood cells lyse, and they release nutrients. So it becomes a really nutrient enriched medium. Okay, for growth. It's diplococci, so you see it in twos. Looks like a coffee bean. And these are the main like valence factors. Now, don't get me wrong, I'm not saying that meningitis or meningitis, whatever, and it does not have IgA protease. It does, it's a general feature, but it's not the main one that's involved in the disease, in the pathogenesis of the disease. So I'm mainly focusing on the virulence factor that's directly related to the pathogenesis. So the fact that it's capsulated causes meningitis, the fact that it has all of these different strains, okay, makes it very hard to control. The fact that uh, its transmission is by air droplets. Keep this in mind, general concept. Anything that's can be like transmits from person to person through air droplets means it's very hard to control in crowded areas. Because in crowded areas, people are coughing and sneezing in really close proximity, so it's very hard to control. That's why this is very common in Hajj. Okay, and we have a special vaccine for that. I'm not sure if it was mentioned or not. I don't remember seeing it. This, yeah, this is very common in Hajj because it's um it's yeah, its transmission is by air droplets. Okay, any disease that's basically transmission by air droplets, its incidence increases a lot in crowded areas. Okay, so meningitis, remember maltose and glucose, M and G. Okay, so it ferments maltose and glucose and it's oxidase positive because, how do we remember it? The brain really needs oxygen supply. It's the first to get damaged when there's no oxygen supply. Okay. Difference in gonorrhea is the main virulence factor is IgA protease. So the main it prefers the mucosal surfaces or membranes. So it causes urinary tract infections, genitourinary infections, and conjunctivitis in newborns. Okay, and it's sexually transmitted, not by air droplets, and it's known for its drug resistance. Okay, so what are the two methods of drug resistance? We said it's it has beta-lactamase and it reduces the permeability of the drug. If you remember, which other organism that's gram-positive also has beta-lactamase? Yeah, of course, exactly. Okay. Any question for the gram-negative oxide? All clear, I'm glad. Okay. So we're more than halfway through the lectures. Inshallah, this won't take too long. Okay. So now for the gram-negative bacilli. How does it look like? This is very obviously pink, there's no contribution of blue whatsoever. So you can tell it's gram-negative, and you can see that they're rods, okay? Another name for bacilli is rods, okay? So you can see if you use different resources, it will either say gram-negative bacilli or gram-negative rods, okay? Same thing. So let's start. So we're done with these, okay, which are the Neisseria. Now we're moving on to the bacilli. So bacilli are classified based on enteric and non-enteric. Okay, so these are non-enteric and these are the enteric ones. Now, you took GI, you know what enteric means. Enteric means it affects the GI. So this is how like we know it, but in reality, enteric means it belongs to the Entericobacter species. Okay, so it's not really that or a genus. Sorry, I forgot like these differences was in them. So this actually means Entericobacter, okay, or Entericobacter, this long thing, okay. This the way we like to remember it as students is that it's the one for the stomach issues, okay, because why? Because there's actually one that does not cause GI symptoms, but it's still under enteric. Okay, so the real reason we call it enteric is because it's gastric, even though it has stomach flu, this one doesn't. Okay, so these are the main causes of stomach flu or gastroenteritis. We're gonna go through them. Now, the way we classify gram-negative um, enteric ones is through lactose. Okay, so again, we have enteric and non-enteric. How do we know, like, how do we classify the enteric ones? Based on lactose fermentation. Okay, so they can either be lactose fermenting or lactose non-fermenting or non-lactose fermenting. Okay, the lactose fermenting is E. coli and Klebsiella, but you guys did not cover this. It's so sad because this is such a cute bacteria. It's very sticky and it looks really cool on agar. Okay, so the fermenting one is E. coli, and non-fermenting is Shigella, Salmonella, and Proteus. Okay, now how can we tell if something is lactose fermenting or non-fermenting? Basically, there's an agar specific to this thing. What is it called? McConkey agar. Okay, so McConkey agar is used to tell if this organism is lactose fermenting or non-lactose fermenting. Okay, which means we call it a differential agar. Okay, so this is the McConkey agar. Basically, it contains lactose. Okay, and if it's positive, if the organism can ferment lactose, it shows up as pink. Okay, and if it cannot, it shows up as yellow. So this is negative and this is positive. Okay, which is the pink. So, okay, this wasn't supposed to appear all at once. So it's okay. So let me give you a scenario of how questions come. So let's start. Try not to read this. Okay. So we have a gram-negative bacilli. It's suspected of causing watery diarrhea. Okay. So we know it's among the enteric ones, the gastroenteritis ones. Okay, we're gonna go into detail, don't worry. It shows up pink on McConkey agar. What could it be? E. coli, exactly. So, because E. coli is lactose fermenting, okay, and lactose fermentation appears pink on McConkey agar, okay. A way I like to remember this is coli sounds like cola, and cola is a drink. So I remember milk. I don't know, just desperate measures, okay? So cola is like cola is a drink. So remember milk has lactose, you know. So remember this one is the lactose fermenting one, okay? Now, what if we cultured the organism and it appeared yellow on McConkey agar? What could it be? Klebsiella for what? Did you say Klebsiella for the previous question or this question? No, if you go back, Klebsiella is actually fermenting, but it's not really taught by Bota. I did not find it in your slides, which is why I'm not mentioning it. But it's actually part of the fermenting. Did you guys take Klebsiella? I don't think you guys took Klebsiella. Maybe I missed it on the slides, I'm not sure. But yeah, so just focus on E. coli. Yes, if it appeared yellow, it means it does not ferment lactose. It's either Shigella or Salmonella. Why did I not say Proteus? Because Proteus does not cause GI symptoms. Okay, so in this specific scenario, if it appeared yellow, then we're suspecting Shigella or Salmonella. Okay, now how can we tell if what we're working with is Shigella or Salmonella? There's actually something called Salmonella Shigella agar, and it's selective for Salmonella and Shigella, so only these two can grow on it. Okay, and basically what happens is Salmonella produces hydrogen sulfide, which appears black. Okay, because it appears black, we're able to tell if we're working with Salmonella or Shigella. So if it appears black, it means it's Salmonella. If nothing happened on this agar, then it's probably Shigella. Okay, so again, uh, let's repeat the scenario. We have a gram-negative bacilli that's suspected of causing an enteric disease. If it appeared pink on McConkey agar, it means it's positive for lactose fermentation. What could it be? E. coli. What if we cultured it on McConkey agar and it was yellow? It means that that's negative. Okay, yellow is a negative test on McConkey agar, which means it's non-lactose fermenting. What could it be? Shigella or Salmonella? How do we differentiate? Again, between Shigella and Salmonella? Exactly, by the hydrogen sulfide production. What's the name of the agar? Yes, SS agar or Salmonella Shigella agar. Okay, so we test for hydrogen sulfide production on Salmonella Shigella agar. No black spots were seen. So what is the organism? Shigella, exactly, because Salmonella is the one that produces hydrogen sulfide. Do you get like how this classification system works? So for the bacilli, it's either enteric or non-enteric. We take the enteric ones, we divide them into lactose fermenting, non-fermenting. Fermenting is E. coli, non-fermenting, we test further for hydrogen sulfide. Okay, so this is like the basic classification system for the enteric group. Okay, now a common point of confusion if you're cramming and everything kind of feels like it's jumbled up is remember the Staph aureus agar? What was it called? Yes, the mannitol salt agar. What color did it appear if it was positive for mannitol fermenting? Yellow, exactly. So please don't confuse this. What's the color of lactose fermenting? Like positive for lactose fermenting? It's pink, exactly. So this is different. Don't confuse these because I remember last year this happened where you're kind of cramming everything and in the exam you see this and you go like, wait, was positive yellow or pink? Because they're both pink and yellow. Okay. So remember the mannitol, we said aureus, it's like an aura. So it's like very luminescent and gold. So yellow is the positive test. For McConkey, it's the pink. Okay. The reason for this is they use different pH indicators, so that's why they're different colors. That's it. Okay. Now, this is basically how we classify them. Okay, so we talked about the classification system. Now we're going to discuss each one in detail. Okay, now let's start with the E. coli. E. coli has two types. Okay, there's the enterotoxigenic E. coli and the enterohemorrhagic E. coli. Okay, enterotoxigenic E. coli just causes traveler's diarrhea or water diarrhea. This is very important. It might give you a hint in questions. Okay, I remember we got this, which is traveler's diarrhea, and it was like which of the following is the causing agent. Okay, so water diarrhea or traveler's diarrhea is the enterotoxigenic E. coli. The enterohemorrhagic coli, from the name hemorrhagic, okay, so it causes bleeding. So this one actually gives you bloody diarrhea. Okay, bloody diarrhea. This is the main difference. This is watery diarrhea. This is bloody diarrhea. This is because it's hemorrhagic. Okay. Now, what's really important to know is this number. Okay. This is O157H7. What does that mean? O and H and even K are different antigens on the surface of the organism. H is actually the antigen that's present for the flagellum, and O is for the polysaccharide chain, and K is for the capsule. It's not written here, isn't it? Okay, so K for capsule, H is for the flagella, O is for the polysaccharide. They're basically present in different proportions on different strains. Okay, so the first strain can have this number of H and this number of O. Another strain can have different, ratios of these antigens. So there's this very specific antigen form or ratio for the enterohemorrhagic E. coli. It's O157H7. Please remember this. Okay, this came as a question for us as well. Um, so this one is responsible for causing bloody diarrhea. Okay, and it's because of consumption of uncooked meat. Okay, now I'm not going to go into too much detail because there's no time. This is like the general concept that you need to know. Okay, again, this is the lactose positive one. Now, Salmonella basically also has two types. We have the typhoidal, we have the non-typhoidal. Typhoidal is the one that causes typhoid fever. The non-typhoidal just causes gastroenteritis and food poisoning. Okay, this is very easy to remember because everyone says like don't eat raw cookie dough, you'll get salmonella. So this is very known for its food poisoning when you consume raw food, like I mean raw eggs, raw chicken, raw meat, if it's not cooked properly. So this is the non-typhoidal strain. Okay. Again, serotype means different strains. Okay. So the non-typhoidal one, gastroenteritis and food poisoning. Typhoidal one is the typhoid fever. Now let's go into more detail for the different types of salmonella. We said the non-typhoid one is the one that causes gastroenteritis or food poisoning, and it's non-invasive. What do we mean by non-invasive? Basically, let me draw it. So uh, yeah, so basically, let's say this is the intestinal wall, okay? Large intestine, or also remember small and large, okay? Just in case. Okay, so this is the intestinal wall, okay? Non-invasive means the bacteria comes and it colonizes here, okay? Sorry, this is the laptop, not my iPad. So it colonizes on the surface, okay? So it causes inflammation of the enterocytes are basically the cells found in the intestinal wall. So it causes inflammation here because it's just infecting the surface. Okay, so inflammation is itis, so gastroenteritis. Okay, invasive means it can actually penetrate, okay, and reach the blood and go to different organs because of the blood flowing. Okay, so the non-invasive one, just very simple. You can remember it stays along the gastrointestinal tract, doesn't really invade anything, doesn't reach the blood, doesn't reach other organs. It's just there colonizing and infecting the surface. Okay, so it just causes inflammation, just gastroenteritis or food poisoning. Okay, how is that the transmission is through consumption of eggs, meat, and poultry that is not cooked properly and through feces. What does this mean? Basically, people who have this disease, okay, and there's poor like sanitation in that area, mainly in poor areas, and the example, there's feces in a body of water, like there's like a pond or something, okay? And there's feces in it that's in fact of an infected person, and then someone comes and washes their hand with this water, okay? They can also get that disease, okay? Um, so the symptoms is nausea, vomiting, non-bloody diarrhea, okay? Why is it non-bloody? Again, because it's non-invasive. It does not invade, it does not irritate, it does not like cause any bleeding, anything that's like to exacerbate, you know? It just causes inflammation along the intestinal walls. So the diarrhea that's produced does not have any blood in it. Okay? Now, for the second type, which is the one that causes typhoid fever, it's very invasive. So again, what did we say invasive means? It means it can actually penetrate through the GI, it can reach the blood. Okay, so you know, bacteria in the blood means sepsis, and because the blood flows, and you know, the blood it supplies different organs, goes different places, so it can actually invade other organs as well. So it causes sepsis, and blood flows everywhere. So on its way, it causes the infection of multiple organs. Okay, this is the difference between invasive and non-invasive. Okay, and a main important thing here is that this is mainly through human to human transmission, not really by eating eggs and poultry and stuff. Okay. And one more thing is that uh, do you guys know what reservoir means? Okay, I'm guessing no. So reservoir means that the organism basically it chooses a place in the body and just sits there and multiplies. It's not really, yes, exactly. It's like a storage. So it's not really actively causing an infection. It just sits there, finds a place, and just multiplies and multiplies and multiplies. Okay, so a person can appear totally normal, okay, but they're actually infected because the organism isn't causing an infection. It's not like actively infecting. It's just sitting there multiplying, 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 and then it can decide to cause an infection. Okay, so the reservoir in the typhoid fever is the gallbladder. Okay, remember this. So you can have asymptomatic carriers because it just sits there and multiplies without even causing the symptom. Okay, so remember the reservoir for the invasive one is the gallbladder. It's very easy to tell the difference between them because one is very non-invasive, very like just inflammation, food related, GI related, vomiting, diarrhea, and the other one reaches the blood, very invasive, can have a reservoir, sits there in the body, you know. So these are the different types of um, salmonella. Okay, don't forget the name. It's typhoid fever. Okay. Now moving on to Shigella. Basically, Shigella is very simple. The only thing I really like have time to go through and is very important in questions is the diarrhea and dysentery. So how will they ask you about Shigella in questions? Basically, they'll mention that the diarrhea started off as watery, and then over the course of a few days, it becomes bloody. Okay, and they'll tell you there was no black formation on the agar, the SS agar. So what could it be? It's Shigella, you know. So this is very easy examination wise, you know. So the next one is Proteus. Now, Proteus is very different than the rest, even though it still lies under the enteric classification, but it does not cause GI symptoms. Okay, it's the only one that does not cause GI symptoms. And basically, uh, the cool thing about Proteus is that it's mobile. Okay, this is not from your lecture, but it's really cool. So I really wanted you to see it, and you probably will see it in the lab. Is that do you see like these swarms that are formed? It's because it's mobile. It moves around. Okay, that's why Proteus looks like this on agar. This is very cool. Now, what you have to remember about Proteus is that it's urease positive. Okay, this is very, very high. You'll probably get a question on this. Proteus is urease positive. Okay, if you want to write it down right now or anything, please remember this. Okay, now this makes it easy to kind of remember the what does it cause because urea you can remember urinary tract. Okay, so urease positive means it can break down urea. When you break down urea, you get ammonia, which is NH3. You don't need to know this, I'm just explaining like what happens. And this ammonia is very basic, okay? Very alkaline. And when you have an alkaline environment, it causes the precipitation of Mg and magnesium and calcium, and you get stone formation, okay? Because these precipitate together. So any question that's going to hint towards Proteus is either going to mention urease or it's going to mention something about kidney stones, okay? Or stones within the urinary tract, okay? Whenever you see stones in a question or urease, think of Proteus, okay? Now, we still did not mention H. pylori. We're going to mention it like um, in a bit in the other lecture, but I want you to remember that these two are urease positive, okay? But this one is for the urinary tract kidney stones, and H. pylori is mainly GI related, okay, like peptic ulcers. So please remember this, okay? Okay, cool. Okay, let's summarize the what we just discussed, which is the enteric ones. Okay, so how do we classify gram-negative enteric bacilli? Enteric. Yes, lactose. So based on lactose fermenting and non-fermenting. Which one is lactose fermenting? Exactly, E. coli. Now, what are the two types of E. coli infections? Yes, so we have enterotoxigenic and enterohemorrhagic. So the enterotoxigenic causes watery or traveler's diarrhea, and the hemorrhagic causes bloody diarrhea. Now, what's the antigen ratio in the enterohemorrhagic one? It's okay if you don't remember it still, but no, I really hope you do. Yeah, 0157H7. Okay, this is very important. So if you get a question about bloody diarrhea and 0157H7, remember it's the enterohemorrhagic E. coli. In our exam, they actually asked us about the number itself. They got us a case of bloody diarrhea, and they were like, which of the following is the antigen proportion? There was like 07H153. It was really close together. So please remember this, okay? 0157H7. Now, so it's basically this, okay? Now, which ones are non-fermenting? We have three non-fermenting. Shigella, Proteus, Salmonella. Exactly. Yes. So Shigella and Salmonella are the GI related and the Proteus is not. Okay. So these three are non-fermenting. Now, how do we differentiate the non-fermenting? Mainly these two. Yes, these Salmonella, Shigella, agar. So how can we tell on the agar? What are we gonna see? Black dots. Okay. Why is it black? Because of production of what? Yes, hydrogen sulfide. Okay, so yes, if it's black, it means it's salmonella. Okay, what are the two types of salmonella infections or diseases? Yes, we have typhoid and we have non-typhoid. So typhoid is the invasive one, which causes sepsis and invades. Let me show you. Yes, typhoid is invasive, which means it can reach the blood and invade organs. Non-typhoid is non-invasive, so it gives you non-bloody diarrhea. So the non-typhoid is the one that's associated with the food poisoning and gastroenteritis, while the typhoid one, it's related to sepsis and yeah, a lot of damage. Okay. Now, which one has a gallbladder reservoir? The typhoid one, exactly. So, yeah, for Shigella, what you need to remember is that it goes from normal diarrhea to dysentery, which means it starts off as non-invasive, then it becomes invasive because you have the blood. Okay. And then, yeah, this is important. I forgot to mention this. Shigella works through the Shiga toxin, okay, which is cytotoxic, which means it's like it kills the cell. Okay. So remember that, very easy to remember Shigella. Okay. What about Proteus? What does Proteus cause? Yes, urinary tract infections. What is mainly what? Like, what does it cause? What will give away in a clinical scenario that we're dealing with Proteus? Yes, exactly. Stones, kidney stones. Yes. So, stone formation is the main giveaway in a question. Okay. Why do we get stone formation? Because it's urease positive. Urease breaks down urea into ammonia, and ammonia is alkaline, so it causes the precipitation of magnesium and calcium. Okay, so that leads to the stone formation. Okay, so remember urease positive and stone formation immediately go to Proteus. Okay,
A lumber yard is a location where lumber and most of the products used in
I'm sorry. Okay, so now we'll move on to the non-enteric ones. Okay, remember it here. Sorry, 1 second. Yeah, so we finished the enteric ones. Okay, we just reviewed them. Now we'll move on to the non-enteric. Non-enteric ones are just these. Okay, okay. Just pretend you didn't see the question. Okay, so the non-enteric ones, I just have them summarized. They're very easy. They're Haemophilus influenza, Bordetella pertussis, Brucella, and Pseudomonas aeruginosa. Now, Haemophilus influenza is transmitted through aerosol, which means through just like the air, and it causes upper respiratory tract infections. From the name influenza, you can kind of remember that, like, when you have influenza, you're coughing and you have, your throat hurts, sore throat. So remember, upper respiratory tract infections is because of H influenza, and because it causes upper respiratory tract infections, you can remember that the virulence factor is IgA protease because that's a mucosal surface. You know, uh, the throat, the airways, all of that, the pharynx, these are all mucosal surfaces, okay? So in an exam, if you do get confused and you want to remember what's the valence factor, which by the way, they don't really ask what is the valence factor, you know what I mean? You might get a couple questions, but it's like they only focus on things that are very important, like capsule, IgA protease, M protein that causes remember the rheumatic heart disease, where the M protein looks like the myosin on the cardiocyte, so it attacks it. So only very, very important things. So if you do get lost, you can remember, okay, if influenza causes upper respiratory tract infections, that is a mucosal surface. So IgA protease has to be right, you know? Um, yeah, أكيد في many others بس you guys memorize them. I feel bad بس no. Okay. Now, each influenza can be encapsulated or non-encapsulated, and this is very important. Okay. Now, remember the chocolate agar that we used for Neisseria? And we said that the chocolate agar is just basically heated up blood, where we heat up blood and the blood cell breaks and it releases nutrients. Okay. What are these nutrients? They're basically X factor and V factor, or in other words, hemin and NAD+. Okay. These are the nutrients that are released that are present in the chocolate agar. And Haemophilus influenza cannot grow without these factors. So we can only grow it on chocolate agar. Okay? So remember this. So for now, like so far we have H influenza and Neisseria or chocolate agar. Okay, that's what I meant when I said it's not really selective because it doesn't inhibit the growth of other organisms. No, it's just a bunch of nutrients where if an organism needs it, it can grow on it, you know? So for H influenza, don't forget the chocolate agar because it requires X factor and V factor for growth. Okay, it cannot grow without it. You cannot grow it on blood agar normally. Okay, it has to be chocolate agar. Now, for like, so these are the main high yield points for H influenza. For Bordetella pertussis, also aerosol, and the disease is called whooping cough. This is high yield. And basically, the virulence factor for this is very unique, where basically it causes agglutination in between the cilia of the respiratory tract. Okay, so there was a picture for this in the lecture. Okay, where it's like agglutination between the cilia. I don't know if you guys remember that. This is very unique to Bordetella pertussis, which is why you need to know it. You know what I mean? Like anything that's very unique to that organism or very related to why that disease happens, you need to remember. You know, not that long list of virulence factors that the doctor lists over and over again. You know, don't worry too much about that. So remember the vaccine that we used for Corynebacterium diphtheria and for Clostridium tetanus? The P was for Bordetella pertussis. So it's the same vaccine that we use. Okay. And basically, the main people that are affected by this, I remember, was unvaccinated children. Okay. So we have to vaccinate people to prevent the pertussis. So these are the high yield points for pertussis. Remember whooping cough and remember DTAP. Remember this special virulence factor that it has. Now, brucella, what you need to remember is that it's zoonotic, which means that it's transferred by animals, okay? Mainly mammals like goats and sheep and stuff like that. Zoonotic does not mean insects, okay? That's what's the name for that? Oh, I forgot. But yeah, zoonotic does not mean insects. It means like animals with vertebrates. Okay, so basically, like actual animals, you know, like goats and sheep and the herd and stuff. So if a question was trying to hint towards brucella, it would be related to farms and herds. Okay, now, yeah. So what does it cause? It causes brucellosis, and the main symptoms of brucellosis is arthralgia, which is pain in the joints, and hepatosplenomegaly, which is enlargement in the spleen and the liver. Okay, I don't think they'll ask you about this. There's no way. This is very like we just took this in heme, you know. So I don't think you guys will really be tested on this just for the sake of including everything. Now for Pseudomonas aeruginosa, this is very important. Why? Because it's very special on agar because it produces a pigment. Okay, it produces pigments like biocyanin. I'll show you a picture. Yeah, this is it. It produces pigments on agar. Okay, and in our exam, I remember they asked us why does it appear green, and the answer was biocyanin. And surprisingly, many people did not know this that we had to appeal the question, which is very weird because this is a very high concept. Okay, so please remember this. The reason it causes this green is because of the piocyanin. Now, some strains of Pseudomonas can even produce bilirubin, which is red, okay? But this is way more important, okay? So if they give you a scenario where, for example, a microbiologist was in the lab and she cultured an organism on a nutrient agar, this see-through agar that's clear, it's just regular nutrient agar, you know? So if she was in the lab, the microbiologist, and she cultured an organism and it produced a really strong smell with green pigments, what could it be? Pseudomonas. I know this sounds like a very stupid question, but questions do come like this because POD is about testing general knowledge. It's not about details. Okay, so do not get lost in the details, please. It's genuinely just about knowing what's what, where's what produces what, what's known for what. You know, it's very general concept. So just have a firm grasp of the high yield concepts, and I promise you'll be okay. Okay, so remember anything that looks like this is Pseudomonas. And what's special about Pseudomonas is that it mainly infects immunocompromised people. What does immunocompromised mean? It means that people who are they just had a blood transplant, so they're on organ transplant, so they're on immunosuppressants. People who underwent chemotherapy, people who are their immunity is very compromised, very low immunity. That's where we tend to see this. Even in every block after POD, you're gonna have micro in every single block. In here, you're gonna have micro about organisms that cause infections in the nervous system. In CVP, you're going to have it related to cardio and pulmonary. Every single block, you'll have micro. And you'll notice this disease is never on the top differentials for infections. It's only on the top differentials if it's an immunocompromised patient. We only think of Pseudomonas being the reason of infection if it's an immunocompromised patient in general. Okay, so these are some virulence factors. It does have a capsule and it has a mucoid capsule. Okay, so it's like a sticky capsule. So just for your knowledge. Okay, let's review the gram-negative now. Which bacteria causes UTIs with alkaline urine? Okay. Before I continue, do you guys have any question regarding gram-negative? Do you want to review it? Do you want to go over something? Do you want me to ask you more questions? Anything? Yes to what? Review. Okay. We can go back to this one. So we talked about in gram-negative the circles and the rods. Okay. For the cocci, we said that it's Neisseria. Okay. Neisseria basically. Okay. What are Neisseria? Let me ask you this. Remember someone who travels?
Okay.
I'm sorry. Yeah. Do you guys remember the agar? It's very important. Anything, make mistakes, it's fine. It'll make you remember it more. Yes, exactly. Thyre Martin. Remember, Martin went to Syria. So Thyre Martin is for Neisseria. Okay. Okay, what's the main valence factor in meningitis? Capsule, exactly, because meningitis. What's a test we can do in the lab that will be positive for Neisseria meningitis? Let me see if you remember this. Yes, oxidase positive, exactly. This is very important to remember. Okay, now, uh, what about gonorrhea? What's the main valence factor here? IgA protease, exactly. So it causes uh, basically urinary tract infections, conjunctivitis, basically mucosal surfaces. Remember, this one is transmitted by air droplets. This one is sexually transmitted disease. Okay, they look like coffee beans. They can grow on they can grow on chocolate agar. The specific agar is Thyre Martin. Okay, then we go to the bacilli. Bacilli is classified based on enteric and non-enteric. And the enteric is further classified based on what? Exactly, based on lactose fermentation. Now, E. coli is lactose fermenting. Salmonella and Shigella and Proteus are lactose non-fermenting. Ignore Klebsiella, okay? It wasn't discussed. Now, to differentiate between Salmonella and Shigella, we said that Salmonella produces hydrogen sulfide on the SS agar, which is black. Now, we already had a question about this, so you guys remember Proteus, okay? Now, for the for the E. coli, we said we have watery diarrhea or traveler's diarrhea, and we had the bloody diarrhea. So enterotoxigenic and enterohemorrhagic. Okay? Enterohemorrhagic is O157H7. For salmonella, we have typhoid, we have non-typhoid. Typhoid is invasive, so it causes sepsis and it can reach organs. Non-typhoid, not really invasive, just joints, just causes gastroenteritis and food poisoning. Okay, so that's the difference between the strains. For Shigella, we just mentioned that it starts as watery diarrhea, then it evolves into dysentery, okay, which is bloody diarrhea. So it starts as non-invasive and continues as invasive, okay? And it does that through the Shiga toxin, okay? Protease, we said we need to remember that it's urease positive and it causes kidney stones, okay? Then we moved on to the non-enteric, which are these four, okay? And then we mentioned the main virulence factors and the diseases that causes, okay? Now I have more questions in the end for this. What is SPP? It means species. Okay, so if I wrote SPP next to something, it means species. Okay, okay, cool. So I hope it's all clear now. Do you guys need more revision? Any questions? Okay, okay. There are more questions in the end where we're going to review everything all together. So inshallah, that will also help refresh your memory. So this is the last part, which is the unconventional organisms. I hate this part so much. There are actually organisms that you guys took that I've never heard of before. So I don't know, your lectures are very, very dense. So, okay, let's start. So the way we classify um, these organisms is based on them being obligate intracellular, which are these, and not obligate intracellular. The way you can remember this is, let me draw this, is rock. Okay, sorry for the handwriting. So obligate intracellular is the O for obligate, C chlamydia, R is rickettsia. Okay, so these are the obligate intracellular. What do we mean by obligate intracellular? It means it needs to be intracellular to grow. It can't survive otherwise, you know? But not obligate intracellular, it means not necessarily. It does like to live intracellularly, but it can survive otherwise. Okay, so before we start with them, basically these are the organisms that have weird cell wall components or weird alterations in the cell wall where they don't respond to the gram staining that we usually do. We can't clearly put them into gram positive and gram negative. You know, that's why they have their own classification system. Okay, because they have certain alterations in the cell wall. We will go through everything, don't worry, where it doesn't allow it to clearly show up as gram positive or gram negative, or even pick up on the stain in the first place. So that's why they're like their own world. Okay, so the way I like to classify them, to remember them, is I put them into the ones that have a weird cell wall, like Mycobacterium, it has mycolic acid, and Chlamydia actually lacks muramic acid. So because these have altered cell walls, they're not the usual cell wall that we have, which is the peptidoglycan and everything. They have alterations in the cell wall. They don't pick up the normal stain, the gram stain that we use. Okay? So these are the ones with the weird cell wall. The ones with an absent cell wall are the ones that are, it's basically mycoplasma. Ureaplasma is a type of mycoplasma. Okay, these don't even have a cell wall in the first place. Okay, that's why it does not pick up on the gram stain. Now, the ones that are caused by bugs, which is rickettsia, the way to remember this is ricket sounds like cricket. Cricket, sarsur. So that's how I remember that rickettsia is caused by the bugs. Okay, this is important. Then it's the spirally ones. Okay, you can either have spirillum, which means that it's basically a normal bacteria, but it has like the spirals attached to it, or spirochetes, where the entire thing is actually spiraled. Okay, so it's the spiral ones. Spirillum is Campylobacter and H. pylori, and spirochetes are Leptospira, Borrelia, and Trichoderma. Now, these are important points to keep in mind. It's that chlamydia and rickettsia are actually weakly gram-negative, so they can appear shwy like reddish. Okay, so they can pick up on the stain a bit. And H. pylori also can appear gram-negative. Okay, so Um, yeah, so let's start with Mycobacterium tuberculosis. Okay, so a way to remember this, personally, I used to confuse, so I used to confuse mycobacteria with mycoplasma, okay, all the time. So a way to remember this is the mycobacterium has B and T. So this is the one that causes tuberculosis. So Mycobacterium tuberculosis. Okay, so basically what's cool about this is that it has mycolic acids in the cell wall. So we call it acid fast. Why? Because it does not get washed away by the acid. Even if we put alcohol, if we put anything, it just does not does not respond to the gram staining. Okay, we call it acid fast. Okay, so basically it has its own agar, which is called Lansstein-Jensen. I'm only going to mention the important things. You don't need to know all of this. Okay, but this is just a very good summary. So you need to know that it's called acid fast, and you need to know that it's Lansstein-Jensen agar. Okay, so this is the agar that we use to identify Mycobacterium tuberculosis. Now, the most important part about TB is its virulence factors. Okay, now, one second. Yeah, so the most important thing to know about TB is that it has a virulence factor called the cord factor. Okay, so where is it? One second. I could have sworn I added a picture. Looking for it. Okay, I think I forgot to add a picture. I can just draw it for you. So in order to understand what this cord factor does, I want you to understand. Okay, so you know how basically if you have a foreign particle or a pathogen, it gets engulfed by the macrophage. Okay, so the macrophage eats it. Okay, and inside the macrophage, this is the macrophage. Okay, so it eats up this pathogen and puts it, basically it becomes inside a phagosome. Okay, so we call this part a phagosome, which contains the phagocytosed organism. Okay, then there's an organelle inside the macrophage called the lysosome. Okay, what happens is that the phagosome fuses with the lysosome to produce a phagolysosome. Okay, once it fuses, this lysosome releases enzymes that destroys the pathogen. Okay, so they fuse into one big thing called the phagolysosome, which destroys the pathogen. Okay, and then what it does is it presents the antigen on its surface. Okay, on the MHC. I think if you studied immuno, this would really make sense. But after it breaks down the pathogen, it takes the antigens from the pathogen, presents it on the surface. Okay, then it goes to the T cells. Okay, it tells them, hey, we have this organism. Can you recognize it? You know, because it presents the antigen on its surface and it goes to the T cell and shows it the antigen. Goes like, see, I have this antigen. This is the pathogen we're dealing with. This cord factor inhibits the fusion of the phagosome with the lysosome. So this entire process cannot happen. Okay, so what happens is this actually hides inside the cell, and we can't even tell it's there. It just hides inside the cells, and mainly it hides inside the macrophages. Okay, so it gets phagocytosed by the macrophage, and then it just sits there. Okay, it just sits there. It doesn't get, uh, there's no phagosome lysosome fusion. It doesn't break it down. It doesn't present the antigen to the T cells. There's no immune reaction. It just sits there. Okay, so this can help you remember that it likes to stay within the macrophages. Okay, now this is an important concept. So remember, the cord factor is what causes this. Okay, so yeah, what else you need to know here? You probably know this more in CVP, but tuberculosis and Mycobacterium tuberculosis is very resistant to drugs. It's multi-drug resistant. This is also an important point. And one last thing that I forgot to mention, that because we said it doesn't take in the normal gram stain, we have a special stain just for the Mycobacterium tuberculosis, and this is it. It's called the Zeal-Nelson stain. Okay, this is very, very high yield. You need to know this really well. What is this called? It's called the Zeal-Nelson stain. Okay, yeah. So if you get this picture, this does not mean gram-positive because it's pink. I mean, this does not mean gram-positive. I mean, this does not mean gram-negative because it's pink. Sorry. PB, okay? Positive group. This does not mean gram-negative because it's pink. No, when you see this picture, this is the Zeal-Nelson stain, and it's used for TB or Mycobacterium tuberculosis. Okay? The agar is Lansteen-Jensen. The stain is Zeal-Nelson. Okay? They kind of rhyme. This, yeah. So it's facultative intracellular. This is very easy to remember because we said it sits in the macrophage and just sits there. Okay, so it likes to be intercellular. It doesn't necessarily have to, but it likes to be intercellular. Okay, so any questions regarding TB? Okay, I hope it's clear. These are like the important points you need to know. Okay, again, what does acid fast means? It means that when we give it a dye, which is mainly the fuchsia dye, remember it was bright pink, and then we wash it with acid, or we wash it with like, we want to wash off the color, it doesn't respond. It doesn't get washed off. It sticks. You know, the word fast means like, when we say fasten your seatbelts, it means like, you're tightly holding on to something. Okay, that's what it means. Okay, so we use the Zeal-Nelson stain, we use Lansteen-Jensen agar. Okay, this is what it looks like. We'll definitely get something about this, whether it's in OSPE or final or midterm. This picture is definitely gonna come. They're gonna ask you like, what's the stain used? Zeal-Nelson. What type of organism? Acid fast, Mycobacteria, tuberculosis. Okay, so moving on to Mycoplasma. Now, Mycoplasma, in order to not confuse it with Mycobacteria, Mycoplasma is the one that causes pneumonia for P, and Mycobacteria has B and T, so tuberculosis. Okay, Mycoplasma just does not even have a cell wall. Okay, so it has a missing cell wall. There's no cell wall in the first place. And a way to remember this is the word plasma, like, kind of gives you a sense, something is very watery or very, it's not something rigid. You know what I mean? So this can remind you, this one has an absent cell wall. So it's very wobbly. Okay, so no cell wall. And basically, the P use it to remember pneumonia. Okay, now this has a very, very, very cool pathogenesis, and it's because of its shape. Its shape can help you remember how it causes disease. So let me show you what happens. Basically, let's say this is the epithelium of the respiratory tract, and you have cilia. You have lots and lots and lots of cilia, right? So what happens is, because of its unique shape, which looks kind of something like this, it can burrow its way between the cilia, so it can reach the respiratory epithelium. Okay? Because usually the cilia is protective, and if like any other pathogen would come, the cilia would protect it. Okay? So this one is really, really, really special to the respiratory epithelium, that it's almost made for it, like the way it's shaped. So it kind of digs its way between the cilia so it can reach directly to the surface of the epithelium. Okay, and it's shown here in this picture, okay, where you can see the cilia. Okay, so C is for cilia. Okay, and you see the bacteria kind of burrowing its way between the cilia so that it can reach the epithelium surface directly. Wants direct contact with the epithelium. Okay, and what it does is it impairs the cilia function. Of course, because it's stuck there in between the cilia aren't able to sweep and move and do their job properly. So this causes the accumulation of fluids and like unwanted things along the respiratory tract, which leads to pneumonia. Okay, because remember we said pneumonia is We didn't say it was in general, pneumonia is basically a lot of fluid in the lungs, okay? And a main reason that causes pneumonia is ciliary impairment. So this directly causes ciliary impairment, okay? And the way it adheres to the respiratory epithelium is through protein P1, okay? So remember P1, it's also called P1 adhesion. So it can kind of remind you that it's used for adhesion, okay? So what else? Also droplet transmission. So this can give you a clue in its incidence increases in crowded areas. Okay. There's also a subtype of mycoplasma called hominis. This causes urogenital diseases. This is very low. Like I've never heard of this before. Never in my life. So, okay. So something unique about it also is that it shows a fried egg appearance. Okay, so if you get fried egg appearance, it's mycoplasma. Now, a way to connect the dots, always remember how it looks like. Okay, the shape. Because the shape will remind you of how it causes disease. And then it will remind you that it causes ciliary impairment. That will remind you of pneumonia. Okay, so remember P1 adhesion. This is a main, really important virulence factor that mycoplasma has. Okay, so remember the P1 adhesion. So is mycoplasma clear? Okay, so again, don't confuse this with mycobacterium. Plasma means like watery, wobbly, so there's no cell wall. Okay, and P4 pneumonia. While in Mycobacterium, oh, in Mycobacterium, remember B and C, so it causes tuberculosis, and it does have a cell wall. It's just altered. It has mycolic acid in it. Okay, that's the difference. It has mycolic acid in the cell wall. That's why it's atypical. This one just has a completely missing cell wall. Okay. Now, ureaplasma is a type of mycoplasma, and this is also the first time I ever hear about this. So, from the name urea, remember it's urease positive. Okay. What other organism is urease positive that we talked about? Causes kidney stones. Proteus, exactly. So, urea, remember urea, urine, UTIs, urinary tract. Okay, it all like goes hand in hand, basically. Now we're moving on to chlamydia. Yes, H. pylori also has urease. That's very true. Okay, for chlamydia, it's obligate intracellular. Where is the pointer? Oh, remember we said rock. So the ones that are obligate intracellular are chlamydia and rickettsia. Okay, now, okay, so basically what this one exists, I think you took this in Hana's lectures, right? Do you guys take elementary and reticulate bodies? I hope you did, because this is very high yield, like especially in Dr. Hana's lectures. Yeah, okay. So make sure you understand these questions will definitely come on. Okay, so anyways, so for chlamydia, we have elementary body and reticulate body. These are basically different states in which the chlamydia exists. Okay, so the elementary body, remember E for extracellular? So it's the one that's outside the cell. Okay, so it's the one outside. You see here, EB for elementary body. It's basically floating around extracellularly, and then it comes and it attaches. Okay, it attaches and infects the macrophage, let's say. Okay, so this one is the infectious form. If they tell you which one is the infectious form, is the elementary body or reticulate, it's the elementary body. Okay, because infectious form means the one that causes infection, the one that invades the organism. Okay, that's why it's the extracellular one. Okay, so this one is the infectious extracellular one. It's gonna come and invade the macrophage, or basically it's gonna get phagocytosed. Okay, and remember when we said when an organism gets phagocytosed, it becomes called a phagosome, right? So this thing here is a phagosome. Okay, then what happens is as soon as it becomes a phagosome, it starts replicating inside. Okay, now it's called intracellular. So it switches form from EB to RB. Okay, so now that it's intracellular and it wants to start replicating and wants to grow, it wants to replicate, it wants to, multiply, it changes form into a reticulate body. Okay, so it starts off as elementary, which is extracellular, infective, becomes phagocytosed. Okay, so when it becomes phagocytosed, it starts to want to multiply and replicate. So what does it do to do that? It changes form. Okay, it becomes a different form, becomes a reticulate body. Okay, so this form is the one that's going to multiply and replicate and condense. Okay, so after it multiplies, replicates, and condenses, it's going to release all of these infective forms again into the blood. So these are EVs. Okay, once it's released, it's called an elementary body. Then the process repeats over and over again. Okay, now this whole bag of chlamydia pathogens, elementary bodies, and reticulate bodies, mainly reticulate, this whole thing, they're called inclusions, okay? And you can see them really clearly under the stains, okay? Do you see these little dots inside? These are all called inclusion bodies, okay? So that's it. And it's basically a sexually transmitted disease, we know this, you know? So these are the main important things. It's obligate intracellular, causes inclusion bodies, and the chlamydia trachomatis, that's the exact name, chlamydia trachomatis, is a sexually transmitted disease. Okay, so any questions on chlamydia? Okay, now for rickettsia. Remember rickettsia, rickett sounds like cricket. So remember this is the one where it's what was the word where it's we just took this last week. We had a final event and I forgot. So it's transmitted by insects, okay, not by mammals so it's not zoonotic, okay. Don't call it zoonotic. Zoonotic means it's transmitted by actual uh, mammals, like any other animal that's not an insect. But, yeah, vector is the word for. Okay, anyways, um, yeah, so the ricket basically is, as we said, sounds like cricket. So it's the parasites of arthropods. Arthropods is basically these exact category of insect. Okay. What it causes is Rocky Mountain fever. I remember this R with R. Okay. And this is what it looks like. So maybe you got you a picture. I'm not sure. We actually did not take this last year in POD. Was okay. Um, Yeah, so in case they get you a picture or any clinical scenario where it's about fleas and ticks and mites and, fever and you have spots on the skin, think of rickettsia. Okay. So again, it's um it's obligate intracellular. I forgot to we mentioned this, but just to remind you, ROC again. The first one that we discussed was chlamydia. This one is rickettsia. These both are obligate intracellular. So you'll find them inside the host cells, as you can see here. See these little dots? You'll find them inside host cells. Okay. So now for the last part, basically we have the spiral organisms. Okay. The spiral organisms can either be spirillum, where as you can see in the picture, it's an organism where you have the spirals attached to it or the flagella attached to it. And you have spirochetes, where the entire thing in itself is spiral. Okay, now starting with spirillum, you have Campylobacter jejuni and H. pylori. Okay, and you need to remember that H. pylori causes peptic ulcers. Okay, this is very important. If you get a question with peptic ulcer, you think of H. pylori. You don't think of the enteric ones that we talked about. Remember the enteric ones, which is what were they? Salmonella, Shigella, and E. coli, all of these cause gastroenteritis, they cause diarrhea, they cause all of these things. There's nothing related to peptic ulcers, okay? All of those enteric ones that we talked about are basically stomach flu. They're called stomach flu, you know? But H. pylori causes peptic ulcers, okay? Now, for the spirochetes, we basically have three types. Yes, we have three types. We have the Leptospira, Borrelia, and Treponema. Again, first time I hear about these. I don't know if I didn't study well or they're actually not there in our lectures. I don't remember. But anyways, just one thing that you need to know is that one of them is hooked and two are not hooked. You can see in this picture, the Borrelia is just straight spiraled. The Treponema is straight spiraled. The Leptospira is spiraled with a hook. Okay, so you really need to remember what disease they each cause. Okay, you can even see here, you see the spiral thing in the middle. Okay, so this is a spirochete. So if you get this picture, you don't think of these two. Okay, it's the entire thing in itself is spiraled. So you immediately go to spirochetes and try to diagnose. Okay, so um, the leptospira causes leptospirosis. Okay, and this one it's on wild animals. Okay, so this is a very important giveaway because let's say they give you this picture, okay, which is the spirochetes, and they give you a scenario that's related to animals, like wild animals. I don't know an example, but you got what I mean. It's not about ticks or mites or any. It's not about human transmission. No, it's just animals. And they give you this picture. You think of leptospira. Okay. For Borrelia, causes Lyme disease. Okay. And this one is transmitted by mites. Okay. So it's very common if you get a scenario in the forest. Okay. And he's wearing shorts, for example, and in the forest. Yeah, we didn't take them, so yeah, I remember. Okay, so if you get a scenario where you're in the forest, okay, and by the way, the questions in POD midterm are very direct, so please like don't be scared about it, okay? It's genuinely like very general knowledge, like main classification, is it Coagulase positive or negative? Where do you find things, So questions are very direct. Don't overstudy, and you'll get a question like someone is in the forest and he's wearing shorts, okay, and he got a bite, okay, and this is how the organism appears on staining, okay. What is it going to be? Borrelia, very easy. Like this is how questions go, So remember that this one is mites mainly, and Triphonema is causes syphilis, and this is actually transmitted by humans. Okay, it's actually transmitted and it's by humans. It's not by ticks or mites or animals, it's by humans. Okay, that's the main differentiation. Now, let's talk about Lyme disease. This is the last slide, by the way. So I just want to talk about this because it is important. Lyme disease has a very special diagnostic feature, which is the target rash. Okay, it looks like um, I forgot what it's called when you shoot arrows out. It's a target, so yeah, target. So it looks like a target. Okay, so dark spot here and darkness here. So this rash is very characteristic for Lyme disease. Okay, and there's an early phase and a late phase. You don't really need to know details. Just know that it's caused by the mites bites. Okay, so remember this picture. Remember this. It starts off as like fever, headache, muscle pain. Then later on, it actually spreads and causes neurological symptoms. Okay, that's what you need to know. It has an early phase and a late phase, and basically, the early phase is just fever and the rash and stuff like that. Late phase involves neurological symptoms. Okay, that's why it's a very severe disease. And yeah, so remember the one that causes Lyme is Borrelia. Okay, Borrelia for Lyme. Treponema is syphilis. Okay, so okay, do you guys have any questions? Any questions at all? Okay, we're gonna solve some. I put in some questions from Dr. Bota so we can like discuss them together. Then in the end, we can do like a very quick revision. I'll like go over the classifications for each bacteria, like gram positive, gram negative, and then these, like very quickly. So let's start. Which of the following is likely to produce coagulase? You can tell me top right, top left, bottom right, bottom left. Okay. Yes, top right. What is this organism? Okay, Staph aureus, exactly. So, quick revision on Staph. We said gram-positive cocci, which are the circles. We can classify them based on catalase. Catalase positive is Staph, catalase negative is Strep. How do we classify Staph further? Coagulase positive, coagulase negative. Positive is aureus. Okay. How do we classify Strep? Based on hemolysis. Okay. So, yes. Which of the following is represented? Okay, someone said C. C, yeah. Yes, exactly. It's Borrelia. You can just use process of elimination. We said the one they're asking for spirocate, not spirillum. The spirillum was the H. pylori and Campylobacter. Okay, so we can cross this out. Mycobacterium is the one with the mycolic acid cell wall, has nothing to do with this. Cross it out. Mycoplasma was the one with the opposite cell wall. Cross this. Salmonella was the enteric gram-negative bacilli. No, we cross it. No, so this one's the only one left. In case, let's say you don't exactly know which one, you can just use elimination. Okay. What is spirillum? It's this. Let me show you. This is spirillum. Spirillum means that it has the flagella attached to it. It's not like spiraled in itself. It just has like this attached to it. Okay, so spiral organisms can either be spirillum or spirocate. Okay, which of the following is associated with ulcers? Okay, here I don't expect you to know the answer, but I expect you to know it by elimination. Okay, so do it by elimination. You'll be able to solve it. Lower right, exactly. Yes. So immediately gastric ulcers, we know it's not the enteric ones. The enteric ones just cause stomach flu, vomiting, gastroenteritis, blah, blah, diarrhea. So it's not the enteric ones. It's not Salmonella and Shigella. Okay. And we know it's not spirochetes because spirochetes is like Lyme disease, syphilis, and um, the leptospirosis something. So it's not really GI related. This one's the only one left, which is H. pylori. Okay. And we said H. pylori can stain on the gram-negative stain because, yeah, it can stain. It's like, looks like a gram-negative bacilli. Okay, what about this? Just remember which one causes skin symptoms. It has skin and it's pneumonic. There are actually, I think, two answers here. Okay, someone said A. I'm glad you said A, so I can clear out a confusion. Oh, never mind. Wait, no, how do I go out? Okay, sorry. So basically, okay, everyone went with D, that's correct. There's also one more correct option. What causes skin conditions? B, exactly, pyogenes. Remember, it starts off with skin rashes and pharyngitis and scarlet fever, and then it can, post-infection, it can develop the antibodies against the M protein, which resemble the myosin protein on the heart cells. So your antibodies start to attack the heart. So you'll get rheumatic heart fever. This is post-infection. But during the infection, it's actually pharyngitis, This is rheumatic heart disease, or not? Yeah. So post-infection can lead to rheumatic heart disease because of the antibodies. And during infection, it leads to erythema, I forgot what the skin condition was called. It caused erythema and skin conditions and pharyngitis and all that. Someone here said A, and please don't confuse epidermidis for, oh, it causes skin infections. Okay, it's good, but like not really because it's normally there on the floor. Okay, so what this does, remember what we mentioned for this? What does this cause? Or like in what situations would we expect it to be there? Yes, it is slimy, so it tends to stick to what? Catheters, exactly. So only think of staph epidermidis as an answer if the question has anything related to catheters or insertion of like um, bodies I mean insertion of foreign bodies into the body, okay? Catheters, um, he has a surgical knee, something like implant, which is metal or something, like mainly he's gonna hint to catheters. That's where you think of epidermidis, okay? You don't think of anything else, okay? And let me make sure of this, but I think it's part of the normal flora. One second. Yes. So a way to remember this is that epidermidis, why did it get its name? It's because it's normally present on the skin. Also, I know Dr. Bota hates the word flora, so I'm just going to say microbiota. So epidermidis is naturally present it's a natural microbiota. It's present on the skin. So if you insert a catheter, you kind of take some of this bacteria along with you with the catheter. So it sticks to the catheter and causes an infection because when it enters the body, it's not where it's supposed to be anymore. So now it's considered an infectious organism. If it's where it's supposed to be, which is the skin, it's not going to cause anything. But if once you enter like a catheter, you're dragging the bacteria with you when you insert the catheter. Now it's not where it's supposed to be. It's in a foreign place. It's not on the skin anymore. So it causes this infection. Okay, that's why it's catheter related because it's normally in the skin and that's where we insert. That's why you need to disinfect really well. Okay, so next question. Is that clear first? Which toxin is it? No, I don't think you need to know that. Like, I don't even know that. Even in the lecture, it just mentions that it causes extracellular, produces slime, which facilitates adherence. That's like the only thing mentioned. Okay, no problem. Is this concept clear? Okay, so what about this question? Top left. Now, why did you think of top left immediately? Yes, lice. And we said ricket sounds like cricket. So anything that's like insect mediated, it's gonna be rickettsia. What is this? I really hope you remember this. From the shape, remember what the shape helps it do? Yes, it's myco what? There are two mycos. Mycoplasma, exactly. So what does it do again? It burrows between the cilia, so it can directly cause infection on the epithelial lining. What's the virulence factor for mycoplasma? What helps it stick to the epithelium? Try to remember. P adhesion, exactly, or P1 protein. Yes, exactly. So once it reaches the epithelium, it causes pneumonia because it causes ciliary impairment. Now the ciliary can't move properly, so it leads to accumulation of stuff, which leads to pneumonia and infections. Okay. Does mycoplasma have a cell wall? No, exactly. What about this? What are they trying to explain in this? Chlamydia, exactly. Mycobacterium. Okay, I see what you mean. Okay, okay, I get what you mean. That's a very smart answer, by the way. Now, the reason why this is not Mycobacterium is because we have the reticulate body and elementary body. These are only in chlamydia. In Mycobacterium, you're referring to the cord factor, which inhibits phagosome and lysosome fusion. So yeah, I think that's, yeah. So it inhibits phagosome and lysosome fusion. That's very true. So it just is able to sit there inside the macrophage. Yes, that's Mycobacterium. Very true. Now, here, that's not what they're describing. They're describing inclusion bodies. Okay, this is special to chlamydia. Any questions regarding this? Okay, let me ask you this question. It's very important. What agar do we use for mycobacterium? Not stain, agar. I think you guys forgot. Try to remember. I want you to try. Yes, Layan Jensen. Please remember this. Okay. This one is used to grow mycobacterium. What stain do we use? This is the agar. What stain do we use? Yes, Zeal Nelson stain. Okay, very true. And remember the picture for mycobacterium. Okay, it's those red lines. Okay, that are very far apart because it only sticks to the fusion. We can't really discolor it. Okay, so yeah, so remember that picture. Last question. Which one is responsible for rheumatic heart disease? Let's solve this one together. Let's try to guess. I think because I didn't explain the pictures, you might get this wrong. Okay, let's solve this one together, step by step. Okay, top left. Yes, exactly. Why did you pick top left? What do we see here? Exactly. This is beta hemolysis, which is bright yellow. This is partial hemolysis because it's greenish. I did not show you guys pictures. So let me actually just show you. Oh, 1 second. Okay, I can show you later. Oh no, okay. So basically, how can you come to this answer? Step by step, okay? Second. Okay, so step by step, okay? So first thing is rheumatic heart disease is caused by what? It's caused by the M protein, remember that? It's when the antibodies against the M protein resembles the myosin on the heart, so the antibodies start to attack the myosin. So it ends up with rheumatic heart disease. You can remember that this one is caused by strep pyogenes, okay? So it's caused by, okay, it's caused by strep pyogenes. You get the point. Okay, strep pyogenes. Now, strep pyogenes, was it alpha hemolytic or beta hemolytic? It was beta hemolytic. The alpha hemolytic ones were pneumonia and viridans. Okay, so we know it's beta hemolytic. Okay, just like one step further, is it group A or group B? It's group A because we memorized in the group B is Agalactia because the galaxy is black. Okay, so it's group A, beta hemolytic, strep pyogenes. Okay, so this is the entire answer. So you look for the beta hemolysis, which is yellow on the blood agar. This is how it looks like. It looks like yellow. Okay, and we said beta hemolysis is complete hemolysis of the blood, which appears as bright yellow. The alpha hemolysis is partial, because remember what remember GPA, so group or alpha hemolytic is partial hemolysis, which is green, GPA. Okay, so it's not this one. Which one was mucoid? Hmm, I actually don't remember. I remember that I know Klebsiella is mucoid, but I don't remember which one is mucoid. Let's see, is it in the transcriptions? Pseudomonas, صح. It was in the table. Let's look through it. Oh, there we go. Mucoid. Okay. So Pseudomonas was the mucoid, okay? Um, okay, what is this here? It says mannitol salt agar, and it's yellow. So does yellow mean positive or negative on mannitol salt? Positive, exactly. What was the other agar where pink was positive? McConkey agar, exactly. Someone said negative. Please remember, don't confuse the mannitol salt with the McConkey agar. In mannitol salt, positive is yellow, which means mannitol fermenting. In McConkey agar, pink means positive, which means lactose fermenting. Okay, please don't confuse these two. And understand these pictures because the doctor likes to get pictures in the exam. He really likes pictures. He will get you a scenario and you need to like link it with the pictures. Okay. So I think we discussed all the pictures. It's just the beta hemolysis I forgot to show it to you guys. And there was one more picture where I wanted to show you about the approach sensitivity. Um, Let me see if I can send it. I can Google it. Okay, so I really want to make sure that you see this. Yes, exactly. I want to explain this to you because it's very important. Okay, so remember how we said to differentiate the alpha hemolytic, we look at aptotion sensitivity. So this dark area is the pathogen. Okay, this capsule in the middle, this capsule in the center, okay, is the antibiotic, which is called aptotion. Okay, now if you see here on the left, there is no clear area, which means the antibiotic was not able to kill the organisms around it. So this means this is aptotion resistant. Okay, ignore this name, this is another type, but think of virulence. Okay, but here the P again is the antibiotic aptotion, and you can see there's this clear area here, which means it was able to kill the organisms around it. This means it's aptotion sensitive, which is strep pneumo. Okay, so this is what it means. If they get you the same question, but instead of aptotion they put bacitracin, which is the B, then bacitracin sensitive. Yeah, bacitracin sensitive is group A, which is pyogenes. So this picture would be pyogenes, and bacitracin resistant would be group B, which is the agalacti. Okay, so this is how you interpret this thing. Okay, now let me show you. Um, I don't know how to reach this. Okay, anyways, I just want to go live. Okay, let me show you the beta and I'm sorry, I don't know how I forgot to put these in your lecture. So I really want to make sure I explain them. Okay, perfect. Do you see this? So this over here is bright yellow. So this is beta hemolysis. This over here is green. Remember the mnemonic GPA. Okay, so alpha is partially mouses, which is green. Okay, so remember, green is alpha, this one is beta. Okay, and always like look up like many pictures on Google because you'll get exposed to different types. Don't just, for example, memorize this because in other pictures maybe it won't be this bright. It might be like a more golden yellow, you know? So in order to be more exposed to different varieties, try to Google more and look at pictures more. Okay, so yeah, does anyone have any questions? Do you guys want to review any concept? Anything whatsoever? Is everything clear? If you have any questions, please do like feel free to send me any text, any email, whatever you need, because I'm like fully done with exams, so I don't mind like spending so much time helping you guys out with whatever you need. So last thing, this will only take you a minute, like it's just one question, you know, will take you like a few seconds. If you guys can just give me your feedback, anything we can improve, anything you liked, anything you disliked, please feel free to leave your feedback. Okay, someone's asking a quick review. Okay, can you end the recording?